Hello everyone. Happy to see all of you here in this new episode of the BioClock Academy lecture series. It's nice to see all the familiar faces, some of new names. My name is Joanna Chiu Oswald, and I'm a postdoctoral researcher in the BioClock Consortium. And together with Hannah Brooks, which is a PhD student in the same consortium, we host the session today. The BioClock Consortium is a Dutch research organization for the one of you which are new now in the room. It has the aim to evaluate different aspects of the biological clock in health, in society, and in nature. And the aim of this lecture series is to introduce you to the basic concepts and to keep you with the proper knowledge from experts in the field. The seminar series takes place every third Wednesday of the month. And we're always happy to see you regularly there. And the time frame of today will be the following. So we'll have a 40-minute talk, followed by 10 to 15 minutes of discussion, so roughly 1 hour, so that we then close at 5 p.m. European time. During the talk, you can write your questions in the chat so that we can discuss them at the end, but you can also unmute at the end and ask the speaker directly. And now I give the floor to Hannah, who will introduce you to our guest speaker today, for whom it's a pretty early start, but we're very excited for your talk.
Yes, so also a warm welcome from me. I'm indeed very excited that I can present you to the speaker of today, Professor Joanna Chiu, who will talk about the molecular basis for seasonal timing in insects. She's a professor at the Department of Entomology and Nematology at the UC Davis, where she also leads her own clock lab. And Joanna actually started off as a plant researcher, but then after her PhD, she switched her molecular genetic focus to animals with a specific interest to circadian biology. And she's still continuing that work today. So with her lab, they investigate the regulation of animal circadian and seasonal rhythms by using a combination of molecular genetics, biochemical, and proteomic approaches. And they do this on a variety of insect species. They also use their knowledge on insects to investigate possible pest controls on invasive species. So very nice work. And I'm just very excited that we will talk about insects today. So please, Joanna, the floor is yours.
Okay. All right. Good afternoon, everyone. Yeah, it's about 7 a.m. in the morning. It's not too bad. So thank you so much for the invitation. And the introduction, and let me share the screen first. Let's see. And I'll remove the faces. Okay. So the screen is good. Everyone can see. All right. Okay, so I really appreciate the opportunity to be here with all of you and present some of the work we've been doing in my lab. So as Hannah was telling you guys, my lab is interested in studying the importance of biological rhythms and how they are regulated by endogenous clocks and timers to adapt to often predictable changes in the organism's environment. So we study chronobiology in the 24-hour time frame, so circadian biology, and these are regulated by circadian clocks. But we are also interested in rhythms with longer time frame, so in particular seasonal biology and seasonal rhythms. So far, we have conducted most of our studies using the Drosophila fly model, which of course has great genetic tools, but we have also started dabbling a little bit in some mouse work to investigate organ-specific regulation. So today, specifically, I will focus on a couple short stories, not to make this too long. First, I want to tell you about the progress we've been making in understanding the role of the insect circadian clock in regulating seasonal biology. So this work was started by my postdoc, Antoine, a while back. And then after Antoine left to return to France, a second postdoc, Sergio, took over when he arrived in 2020 from Chile in the middle of the pandemic. And so I'm going to talk about that story first. Then I'm going to dive more into a second story to specifically focus on alternative splicing as a mechanism that mediates temperature-dependent changes in biological rhythms, which is also, of course, relevant to seasonal adaptations. And this project is spearheaded by another postdoc, which actually he was my grad student and now a postdoc, Yao Chai. So let's start with the first story. So seasonal adaptations are critical, of course, for the survival of animals. So many animals, including insects, exhibit seasonal changes in behavior, for example, long-distance migration in the monarch butterfly. Metabolism, for example, in hibernating animals, the metabolism is different in the winter. And of course, there's also seasonal adaptations in reproductive status. So in insects, for example, normally, Drosophila normally undergo reproductive dormancy in the cold winter months. And the seasonal time in animals receive, of course, seasonal cues to regulate seasonal adaptations, as you can see here on the slides. The most notable seasonal cue is photoperiod, but we know that, of course, temperature is also important. There is still quite a bit of unknowns about the seasonal timer. It has long been suggested that the circadian clock actually is involved in measuring the length of the night or the length of the day, but how it's doing that and how it's interacting with the neurocrine system to regulate seasonal physiology and behavior is still a little bit unclear. So let's see. So in 2020, Antoine published a paper showing that a rhythmically expressed protein expressed in the insulin-producing cells in the fly brain, which is here. This protein is called izapsin or Aya. It can respond to photoperiod and temperature and functions to regulate changes in reproductive status in flies. So, this is indicated by the ovary, mature ovary versus ovary without eggs. So when the ovary is dormant, it doesn't have eggs in there. And then when it's actually active reproductively, it has eggs. It's really clear when you dissect the ovaries. So we were pretty excited about this finding, given that Aya has been shown by a number of groups in mammals and birds to have similar functions. And its expression, it actually can respond also similarly to photoperiod. They didn't really look at temperature, but at least for photoperiod, which is one of the key seasonal cue, it responds to that. With the tools we have in fly genetics, we were able to clearly show that Aya expression in the insulin-producing cells in the fly brain is required to trigger winter reproductive dormancy, so a particular winter phenotype. We showed that Aya level is high on the right-hand side in simulated winter conditions, so short photoperiod and cold temperature. Not only that, but it actually peaks at nighttime. So this leads to reproductive arrest. On the other hand, in simulated summer condition, which is on the left-hand side here on the screen, however, Aya level it's much lower. It also shift its peak so that now it peaks in the light period. And this leads to active reproduction in flies. You can see that in the ovary, there are tons of eggs right there. So after the publication of this paper by Antoine, the immediate question that we want to ask and comes to mind is that how does the circadian clock actually regulate seasonal changes in Aya expression, like Aya, especially since that Aya is a rhythmic gene. So how does the clock actually regulate Aya expression, leading to seasonal adaptations? So a couple of published papers at that time already gave us a lot of clues. So these two papers in PLOS Genetics, one by the Yamamoto lab in 2018, and a collaborative work by Bambos, Shaletta, and Rudy Koster in 2019, gave us a big clue that maybe the circadian neuropeptide, pigment dispersing factor, or PDF, it's involved. And PDF may mediate the signal from the circadian clock to Aya. So at this point, my postdoc, Sergio, took over the project. And given these results already published, I would not go through them in details, but we'll just summarize them for you guys using this schematic on the screen. So Sergio found that PDF level in clock neurons, just like Aya in the fly brain, can respond to photoperiod and temperature. So specifically in simulated summer condition, PDF level in clock neurons is really high. So this activates PDF signaling in insulin-producing cells, leading to what is shown down here, protein kinase A dependent phosphorylation of AYA, triggering its degradation. So as we have shown previously, the low level of AYA induces summer phenotypes. But on the other hand, in simulated winter conditions, so PDF level, it's much lower, in clock neurons is much lower, leading to more stable and higher level of AYA protein without the pKa phosphorylation and dependent degradation. So this promotes winter phenotypes such as reproductive dormancy. So just to summarize this figure, so PDF level, it's sensitive to photoperiod and temperature, just like AYA. And PDF is actually upstream of AYA. It modulates AYA protein abundance in the insulin-producing cells, which is a region of the brain that is really key to seasonal regulation in insects. And this is through pKa-dependent phosphorylation and degradation. So, well, then the next obvious question that Sergio wants to address after this paper is how does the molecular clock, which is upstream of PDF, regulate PDF levels in the context of seasonal adaptations? So specifically, Sergio wants to investigate how does the molecular clock integrates the two key seasonal cues, photoperiod and temperature, and how does the clock proteins actually interpret these signals? So how clock proteins interpret photoperiod or light dark signal may be more straightforward, given many clock proteins alter their expression in response to light. For example, the fly clock protein timeless, which is replaced by cryptochrome in the mammalian clock, it's actually light sensitive. So TIM is light sensitive. And it's actually a perfect protein to measure the length of the night. On the other hand, how clock genes or clock proteins interpret temperature, it's less obvious. Except we do know that clock genes such as period and timeless actually exhibit temperature-dependent alternative splicing. So Sergio is particularly interested in testing the hypothesis about timeless, that timeless is critical for integrating photoperiod and temperature cues to regulate seasonality. So we think that TIM, or timeless, short form is TIM, it's the perfect integrator of both of these seasonal cues because TIM mRNA can exhibit temperature-dependent alternative splicing, while the canonical TIM-L protein, which is actually around 1400 amino acid long, which is expressed at 25 degrees, it's known to be light sensitive. So for TIM splicing at the mRNA level, we can see that at warm temperature, you have the canonical TIM-L. In cold temperature, you actually has much shorter version. The most dominant one is actually this one, TIM short cold, which is almost just it's less than 1,000 amino acid long. So obviously, we expect that the function of this protein would be very different. So to start looking at this, Sergio had to do in the beginning is to develop a new antibody because we needed to look at TIM expression in summer versus winter conditions to see if the expression is at all different. So we developed an antibody with an N-terminal antigen so that we can detect both the TIM long isoform and the TIM short co isoform all at once. So as you can see, this Western blot, this is just wild type flies. Genotype doesn't really matter. It's W118. You can see that at 25 degrees, you see this regular canonical TIM-L protein, which is about 1400 amino acid. And at 10 degrees, which is much colder, you can now see that there is increased amount of TIM short cold, but much less TIM-L. And before, I think Sebastian Katner's lab actually showed that TIM-SC is expressed, but it was predicted that it was actually not expressed at the protein level because there was no, because the protein is short and they were not able to detect it using the antibody. But, with the new antibody, we were able to actually detect it, which is great. let's see. Sergio then used this antibody to assay TIM expression in simulated summer versus winter condition over 24 hours. So collecting samples from fly heads every 4 hours. So CT 0, 4, 8, 12, 16, 20. So 4 hours apart. So at summer condition expected, TIM-L is dominant. So this is TIM-L right here. And it shows a prototypical rhythmic expression profile being more stable at nighttime, given that it's light sensitive. On the other hand, in winter conditions, so short photo period and 10 degree, TIM short cold, it's actually dominant. So as you can see down here. And not only that, but it appears that it's much less light sensitive. It's actually present so almost like throughout the day, showing continuous expression the entire 24 hours, even though there is slight probably rhythmicity there. Okay, then the next obvious question is whether the different TIM isoforms, so TIM-L and TIM-SC, can differentially regulate PDF in summer versus winter. Is TIM a major mediator of season regulation? That's the key question. So to address these questions, Sergio and my grad student Rodrigo actually generated transgenic flies expressing only TIM-L or TIM-SC and used those constructs to rescue TIM-L mutant flies. So this Western blot down here just validates the expression of the two isoforms in the two fly lines, so TIM-L and TIM-SC. So these are unpublished data. Sergio is actually working on wrapping up this paper, so hopefully this will be published soon. He's also starting his new lab soon in Washington State University, so he has to finish. Okay, so given the difference in length of TIM-L and TIM-SC, so it is not hard to imagine that the two proteins might function differently, right? So they're missing, the TIM short cold is missing almost like 500 amino acid. So this is immunohistochemistry data showing on this slide, showing that TIM-SC appears to have disruption in nuclear localization in clock neurons. So it's shown here, while TIM-L proteins do translocate to the nucleus as expected in evening time points. On the other hand, TIM short cold, which is on the right here, appears to be more diffuse. You can't really see it goes into the nucleus as in TIM long. It's kind of diffuse and not really going into the nucleus. So this is just a graphical form of the result showing the 3 nighttime points and nuclear localization of red, which is TIM-L expressed in the summertime, and then cold blue here. So we are characterizing the difference of these isoforms further and in terms of their interactions with PER and also, of course, their molecular function right now. But for today, the key question really is whether PDF is differentially regulated by the two TIM isoforms. TIM-L, which is more prominent in summer condition, and TIM short cold, which is more dominant in winter condition. So to answer this question, Sergio performed immunohistochemistry to detect PDF in clock neurons. So in TIM-L versus TIM-SC flies at just 25 degree over 24 hours. So this is his data, and this is actually graphical format on the right-hand side showing all 6 time points. But just for simplicity's sake, I'm only taking out some images showing 2 time points in panel A right here. So he's very excited to see that TIM-SC flies, which is down at the bottom, have low winter-like PDF levels as we expected. So these are actually clock neuron projections, and this level is low in TIM-SC flies, but the levels do not appear to be cycling here no difference between CT 12 and 24, so the daytime point and the nighttime point. On the other hand, in TIM-L rescue flies, you see a rhythmic PDF expression, and you see it actually goes higher at the CT 24 time point. So now the final question for this story is, does this decrease in PDF in winter correlate with winter phenotypes in TIM-SC flies, correlate with winter phenotypes? So the easy experiment to do is just dissect their ovaries and just look at the count basically to see if they have eggs and check their reproductive status. So he did that experiment at 25 degrees. So here he reared flies at 25 degree, long photo period, so summer conditions. So what happens is that, as you can see, graphical format on the right-hand side, Tim L rescue flies have really low level of dormancy, not surprising, and the figure is shown here. On the other hand, Tim SC fly, even though it's in summer condition, reared in summer condition, they still actually have reproductive dormancy, which really indicates that Tim SC is linked to winter phenotypes. So he was really excited to see that. So to summarize this first part of the talk, we found that timeless is an excellent integrator of photoperiod and temperature cues. Photoperiod and temperature were able to differentially regulate TIM isoforms expression. And we showed that these TIM isoforms can then differentially regulate PDF levels. Specifically, TIM SC is associated with low PDF levels and winter phenotypes. So given temperature has such dramatic effect on TIM splicing and its function in the context of seasonal regulation, we actually think that it is highly likely that splicing of other clock components may provide additional seasonal regulation. So in the second part of my talk, let me check the time here, 25th, we're still good. Okay, so in the second part of my talk, I will dive a little bit deeper into our research in understanding how alternative splicing represents a key mechanism in regulating temperature-dependent changes in daily biological rhythm over the calendar year. So for most of us who study circadian clocks, the majority of our experiments have been conducted in constant temperature. For example, in insects, we use 25 degree all the time, which does not totally mirror clocks in nature, in the wild, right? So temperature not only oscillates, especially in temperate region over 24 hours, it could also change significantly over the calendar year. So this, of course, alters the profile of daily behavioral rhythms, which is a seasonal adaptation. So now that we have a better grasp of the basic mechanisms regulating the molecular clock, we think this would be a good time to really start to learn more about how temperature, which changes in nature, could modulate daily rhythms. And of course, this is very relevant to understanding seasonal biology. So this on this slide right here, so panel A shows an experiment by actually Colin Pittendry published in 1954, illustrating the concept of temperature compensation of the circadian clock. So he showed that at a temperature range between 16 degree to 26 degree, Drosophila flies have a pretty robust circadian eclosion rhythm. So eclosion, for those non-insect people, it's when adult flies actually emerge from the pupal case. And this usually happens every 24 hours at around dawn. So this process is regulated by the circadian clock with a roughly 24-hour period length. Despite the change in temperature in these three experiments, these three series, the period length is quite robust. So they keep emerging near dawn. So this reflects temperature compensation. So however, the fact that the period length of the circadian rhythm is robust over a range of temperature does not mean that the profile of that behavior cannot be different. So as you can see in panel B right here, so this experiment also, from the 1960s, using garter snake measures daily activity level over a range of temperature from 21 degree roughly to about 35 degree. So whereas the snakes are more active during the middle of the day at cooler temperatures at 21 degree, they usually are more active at dawn and dusk as the temperature gets hotter and hotter. It's natural for them to try to avoid the hot day in the middle of the sun to survive. So this means, in this case, temperature is an important signal for them to adjust their daily rhythm for seasonal adaptations. So I have already told you about the key role played by the clock gene timeless in Drosophila seasonal adaptations in the first part of my talk. So in addition to regulating reproductive dormancy, which I showed you earlier, TIM isoforms also produce alternative splicing that can regulate other seasonal phenotypes, for example, locomotor activity, daily locomotor activity. So I'm not going to go deeper into this because I already mentioned this. I would be remiss if I do not mention this work by my postdoctoral advisor, Isaac Etheries, in temperature-sensitive alternative splicing of the period gene. He showed actually in 1990s that it's also important for seasonal adaptation for per splicing. It's also important for seasonal adaptation. He found that at high temperature, an intron in the 3' UTR of the period gene is retained. On the other hand, this intron is spliced out at colder temperature, and this leads to more stable mRNA, advancing the phase of both the per mRNA and per protein in the cold temperature, and this leads to advanced evening activity. So, the flies will actually perform all its activities before the temperature gets too cold. So based on what we know about PER and TIM temperature-sensitive splicing, it's possible that splicing of other clock genes could also be similarly regulated, but it has never been looked at. So it's even possible we think that this goes beyond just the molecular clock genes. And we have started to test this hypothesis the last few years. So I'm going to go in the remaining of the talk, first discuss some published results. We just recently published it at the end of last year on splicing of the clock gene, the main transcriptional regulator. And then in the end, I will show you some unpublished data indicating that temperature-sensitive alternative splicing affects really the broader transcriptome, so which is quite exciting for us. So let's start with just splicing of clock and how that's important. So when the clock gene was first cloned by multiple labs, there seemed to be two different splice variants. So there is a slightly shorter one that codes for a protein that's missing 4 amino acid adjacent to the BHLH DNA binding domain, and the longer one cloned in Isaac's lab. So when I look back at those papers, they mentioned that the 4 amino acid difference was likely a cloning artifact. My graduate student and now postdoc, Yao, decided to look into this further, and he saw that actually both isoforms, both splice variants are actually expressed based on a data set from Rauschbach's lab in sequencing data for clock neurons. So Yao then hypothesized that alternative splicing of this clock gene at this location, because of the alternative use of 3' splice site, may be temperature sensitive. He tested this first using just simple qPCR using variant-specific primers, and he found that indeed the shorter splice variant, it's actually, which he termed clock cold, is more prominent at cold temperature. So the name, I guess, fits really well. Yao then asked some very obvious question. First, given clock protein, it's the primary transcriptional activator of clock genes. There's clock cold and clock long encode proteins that have different transcriptional activity. So to answer this question, and he actually did this luciferase experiment reporter assay in S2 cells. So he transfected both of these isoforms into Drosophila tissue culture and just used this assay to measure transcriptional activity. And he saw that indeed clock cold actually has a higher transcriptional activity compared to clock long. He then looked in flies to see if this matches what is observed in flies at warmer temperature, 25 degree versus 10 degree, which is colder temperature. He looks at both clock target and non-clock target using qPCR. And he found that over the circadian day, 24 hours, clock targets are more highly expressed, which is not the case with non-clock target. And this totally matches what he sees in tissue culture experiments. He then asked if this higher transcriptional activity for clock code is due to higher level of clock code binding to the DNA He did this by using chromatin IP, using clock chromatin IP, followed by qPCR, just looking at binding to a clock gene promoter. In this case, he's showing the data for frill. He saw that he did this in both 25-degree and 10-degree, and he found indeed higher clock binding to this clock gene promoter, but only at 10-degree, but only in the early day. So we'll come back to explain the relevance of the early morning difference, but he concludes using just this data that this could explain the higher clock transcriptional output at colder temperature. So now Yao wonders what exactly led to the higher clock code binding to DNA. So looking more closely at the two clock isoforms and the 4 amino acid difference, we were very excited because we're a phosphorylation lab. We saw that there is actually a serine within that 4 amino acid that is missing in clock code. So we immediately hypothesized that this serine could be phosphorylated. And having a phosphoserine, or as you can imagine, a negative charge right next to the BHLH DNA binding domain would certainly disrupt clock DNA interaction. So this could explain why clock code, which does not have the phosphoserine, would bind to DNA more and have higher transcriptional output. So there are other published data that provides justification to our hypothesis. First, it's been shown by Paul Hardin and other labs that time of day dependent phosphorylation of clock is known to regulate its transcriptional activity. So, and second, a long-standing model developed again by Paul Hardin suggests that PER might act as a scaffold to bring in unknown kinase to the nucleus to phosphorylate clock. And that might be how it's dislodging it from the DNA to terminate transcriptional activation. So far, neither the kinase nor the clock phosphorylation site in Paul's model has been found. So could it be serine 13? So inspecting the sequence motif around this area, so Yao determined that there are 2 kinases that best fit this motif and could potentially be that unknown kinase that phosphorylates serine 13. So one of them is CK1 alpha, and the other one is Shaggy. So he tested CK1 alpha. And he did this by actually expressing clock in the presence of CK1 alpha in Drosophila S2 cells and just looking at mobility shift. And he saw that when he co-expressed clock with CK1 alpha wild type, he does see a mobility shift. But when he actually expressed clock with the kinase that version of the kinase, he didn't see a shift. So this results show that CK1 alpha, it's possibly the kinase for serine 13 and definitely a kinase for clock in general. So we then purify clock proteins in Drosophila tissue culture cells expressing both clock and the kinase and use mass spectrometry to identify CK1 alpha dependent clock phosphorylation sites. There were actually 4 different sites that we identified, and Yao was very happy that one of them was actually serine 13, amazingly. So we actually further confirmed serine 13 is phosphorylated in vivo by developing a phosphospecific antibody, which happens to show that serine 13 is phosphorylated only in the early morning time points. And this also explains why the clock chip data I showed earlier at 25-degree versus 10-degree only shows a difference of clock DNA binding in the early day. So after Yao confirmed S13 is indeed phosphorylated, he wants to determine if phosphorylation of this serine in fact affects clock's transcription activity. So kind of backtracking and checking. He generated 2 clock mutants constructs, one encoding non-phosphorylatable serine 13 mutant, and the other one is a phosphomimetic. He transfected these into Drosophila cell culture and used, again, the per-Luck reporter assay to show that indeed the non-phosphorylatable S13A mutant, which sort of it's equivalent to clock in the cold clock code, which is missing the serine 13, actually have higher transcriptional activity. So the non-phosphorylatable S13A actually is similar to clock out, which is as we expected. So finally, since there is a published mammalian clock crystal structure, we were also able to model the S13 residue onto that structure, which give us even more confidence that serine 13 phosphorylation would disrupt clock DNA binding. So we finally confirmed this using clock wild-type protein and serine 13 phosphomimetic proteins purified from E. coli, followed by in vitro assay of biolayer interferometry just to look at binding. Here you can see that this is the serine 13D, just shows, much less binding than the wild-type corresponding protein. I'm not going to go into the data, but finally, Yao also generated transgenic flies to express either S13A or S13D phosphomimetic to support our model and show that S13 is indeed important for temperature responses. So we actually look at the flies in different temperature and show that temperature compensation as well as temperature responses are disrupted. So to summarize, I describe a mechanism by which temperature can modulate circadian output by alternative splicing of the main transcriptional regulator clock. At warmer temperature, you have more clock long. Clock code is also expressed, but I just didn't show it in the model for simplicity's sake. This means that serine 13 can be phosphorylated and clock DNA interaction is reduced. On the other hand, when temperature is colder, it's expected that there would be more clock cold. The overall effect is lower impact of the inhibitory effect of the S13 phosphorylation. So as a result, this leads to elevated mRNA expression of clock targets under lower temperature. So we're excited about this work. In addition, of course, we identified the kinase that regulate, that PER brings in to regulate clock transcriptional activity. And we're excited also because this highlights the coordination of alternative splicing and also phosphorylation in regulating response of clocks to temperature changes. So to end today's talk, let's see, time, 39, which is perfect. Okay, so I want to provide some food for thought. So here are a couple questions we have been thinking about. One is, does temperature-induced alternative splicing also occur in non-clock genes? And really, is this a more broad phenomenon important to regulating seasonal biology in general? So we have started looking into this by using PacBio isoform sequencing. So on a Drosophila glia cell line, we didn't really want to do this isoform sequencing, which requires really high coverage, high depth to identify splice forms using whole heads as starting materials, because there's going to be a mixture of tissues and cells in there. So we actually decided to use this glia cell line, which is light-driven, has light-driven rhythm. And so we can actually just put them in tissue culture conditions and just generate different time points and do isoform sequencing that way. So we did that. We have our first set of data, and we collected glia cells at 2 time points, just CT 12 and 24. But we also did the experiment at 2 temperatures, 25 degree and 12 degree. So panel A shows the Venn diagram of the different genes that are differentially spliced in these various conditions and the overlap between them. So you can see that orange is actually genes that are differentially spliced and at 25 degree versus 12 degree at time point 12. And then purple is actually the differentially spliced genes that are actually at CT 24 and between the 2 temperatures. And you can already see that there's actually a significant amount of genes that are differentially spliced in response to the temperature change, which is really exciting. Correspondingly, there's actually much less in terms of changes when you look at constant temperature at 25 degree, but the 2 time points. So panel B shows the different types of differential splicing, including intron retention, exon skipping, alternative usage of 3' splice site and 5' splice site, so on and so forth. And it's interesting to see that there's a significant more intron retention when the temperature is colder. Not just that, but there's other types of differential splicing as well. So we're really excited to just sort of diving deeper into this data. We're still analyzing this data, but looking at just the co-enrichment of differentially spliced genes, we're excited to find some interesting patterns. For example, in panel A, this is for molecular function of genes that are differentially spliced in response to temperature. We actually see highly enriched categories are kinase activities, which is of interest to us. And there are also other, categories that regulate, transcription factor binding, mRNA binding, et cetera, et cetera. So this is only the beginning. So this actually reinforces our observation that, alternative splicing and phosphorregulation may be intertwined and important for seasonal biology. So we're really excited to actually dive into this further. Oh, sorry. So to end the talk, I would just again like to acknowledge everyone who did the work. So especially Sergio, who is working on seasonal biology, and Yao, who's more into molecular clock and more biochemistry, but he's also interested in the temperature responses, and he's working on the isoform sequencing as well. Also, thanks to our collaborators who helped us with mass spec, isoform sequencing, BLI, et cetera. And finally, of course, we very much appreciate the funding by the NIH. So I'm happy to take any questions you might have. I think this is the end of my talk. Yes.